Alrighty, hi guys. So this is the second part of the week, three video sequence here. So it's the DNA replication part 2 video entitled PCR and amplified products. The idea behind this one is to help explain a bit of the readings. A lot of people seem to struggle with PCR, how it works, the steps behind PCR, and amplified products, what they are, how to determine their length, things like that that you need to know for your homework. So that's the purpose of this video, is to go specifically over PCR and amplified products. The part 1 video went over the actual process of cellular DNA replication, which is a little different. So if you're not feeling comfortable there, you should definitely go and watch that video, and it will help you out there, hopefully. All right, so first off, let's start out by defining PCR. PCR is an acronym that stands for polymerase chain reaction. That's great. Let's go over what each of these parts mean. The polymerase part, if you remember from the DNA replication in the cell, a polymerase is an enzyme that makes long chains of repeating subunits. In the case of DNA replication, the polymerase enzyme we use is called DNA polymerase. So specifically, this term polymerase in the name is referring to that this reaction here, this process, uses a DNA polymerase enzyme, specifically a DNA polymerase 3 enzyme. Now, if you remember from the DNA replication in the cell, the job of DNA polymerase 3 was to read the template DNA strand and build a new strand that's complementary to it. So we're going to use that same enzyme over again, though it's going to have a twist, and I'll tell you about the twist here in a little bit. So that's what the polymerase part of the name means. The chain reaction part, if any of you have any background in chemistry at all, you'll probably recognize this term. A chain reaction is any chemical reaction that once started automatically continues to happen over and over again until it's stopped. And it can be stopped by, there could be a signal that tells it to stop, or we run out of whatever it's using to make the chemical reaction happen. That can stop things. Somehow we stop it. So it's a chemical reaction that once started automatically continues to happen over and over again until it's stopped somehow. So basically, if you put it all together, polymerase chain reaction is a process that uses the DNA polymerase 3 enzymes to cause a chain reaction to happen. Specifically, a chain reaction of linking DNA nucleotides together in a long chain, which is exactly what we did in the DNA replication in the cell. It's just building a new strand. So basically, we're using DNA polymerase 3 to build a new strand. So then what's the difference between PCR and cellular DNA replication? PCR happens in a test tube. That's the main difference, is cellular DNA replication happens in a cell, and PCR happens outside of a cell. It happens inside a test tube. So basically, we're trying to replicate what the cell's doing with the DNA without having to deal with a cell, because cells can be finicky at best. Whereas if you take the process out of the cell, you're now in control of when it happens. And so there's a lot more productivity with it. And we'll go into the uses here in a sec. So basically what polymerase chain reaction is it allows us to collect a DNA sample. So we take a sample of DNA collected from somewhere, and we'll go over some places you can get it. Find a specific DNA sequence that you're looking for on that, and create thousands of copies of that specific DNA sequence. So it's kind of like we're using it as a DNA Xerox machine. We're finding the one page in the textbook that matters and printing off 1 million copies of it, is essentially what we're doing. Now, what do we use this for? Because it's great that we can do this, but a lot of students ask, well, what's the point? Because the readings don't go into a huge amount of detail this week as to what the point of PCR is. We're just they focus mostly on how it works. So it turns out the PCR is actually extremely useful. It's one of the, if not the most, it's one of the most used processes in the entire biology and medical fields. Like when virtually everything we've discovered about DNA sequences and everything like that, genes, a lot of it has come since the discovery of PCR. This has completely revolutionized the entire industry, the entire science. It's been magnificent. Let me show you a couple of the reasons why that we can use it for. Next week, we will learn about how you can use PCR for forensic analysis. This allows you, we can use PCR as a precursor step. Next week, we'll learn about another process called gel electrophoresis. And between PCR and gel electrophoresis, you can take a DNA sample from a crime scene, like from a blood sample or spit or semen from a rape scene or something like that. And you can run PCR and gel electrophoresis on it and figure out who the bad guy was. That's really helpful. With genetic engineering, we use it. We're going to go over that in week 10 as to why you would need to do this. But basically, we can use PCR to make a ton of copies of a gene segment that I'm trying to put into a new creature to make them have a different property. And so I make a ton of those copies and then put it into the new creature and it's able to work. So I don't have to have a ton of copies originally. I can take just a few copies, make a ton of copies, and then put it into the creature because I'll have enough to do that. Scientists use it to study genes. We like to know how long they are. We like to know the sequence of them. Alternatives on PCR, they modify it ever so slightly, but that's how they sequence DNA. So when you send off your, when you do like 23andMe or Ancestry.com and you send off your DNA sample to them and they send you back saying where you're from or whatnot, what they did was PCR. They did PCR on your DNA sample, determined the sequence from the PCR process, and then compared it to their records of DNA sequences of other people around the world and determined which ones you were closest to. That's what they're doing, is PCR. Paternity testing. So again, with the Ancestry.com and stuff, there are some times, like, I don't know if you've ever seen the movie, oh shoot, what's it called? Mamma Mia. In Mamma Mia, they don't know who the dad is. They've got a daughter, and the mom was very promiscuous, and within like a week got in trouble with 3 different guys, and had a daughter, but she doesn't know which of the 3 guys was the dad. And that's the whole point of the movie, is they go through and she eventually just accepts that all 3 of them are her dad. Well, with PCR, with the invention of PCR, they could go in and figure out which of those 3 guys are her dad definitively, which would throw the whole plot of Mamma Mia out the window. Shoot. Medical disease screening. We'll learn in week 9 that you can use PCR and other processes that are based on PCR to determine if you have a genetic disease. For example, it can help you discover if you have sickle cell anemia or hemophilia or a bunch of other things like that. PCR and a bunch of connected processes we'll learn about in week 9 can help doctors determine if you have genetic diseases or not. Virus detection. It's sometimes difficult to determine if you have a virus. Recently, we've had that COVID-19 pandemic. And the idea behind when they do stick that obnoxious swab up your nose, clear back and poke your brain with it kind of thing, like, and draw out some stuff, and then they send it off to some lab. The lab is doing PCR to see, they're looking for a specific sequence of, or a specific DNA sequence that is found in the COVID-19 virus, that, and if it's present in your body, they know that you've been infected with the virus. And if it's not present, then they know you haven't. So they use PCR to detect if you've been infected with viruses, and COVID's not the only one. They do that with a lot of them. And the list could just keep going on and on and on and on and on. But there's a bunch of examples right there that show that this process is extremely valuable to biology. We really aren't very good at functioning without it. So hopefully that helps perk your curiosity as to why this is important, and you're not just learning something because we told you to. All right, let's go over now. Let's go over and talk through how the actual process works. So now you've got all the background and you're probably sick of me talking. Let's show you visuals. Let's show you how this works. And I'm going to switch over to my polymerase, or to my PCR simulator to show you how this works. Okay, so here's my PCR simulator. I know it's crude, but it'll get the idea across. It's a spreadsheet because I only know how to build in spreadsheets, and I love spreadsheets. So yeah, hopefully this will help. What we've got across here, this is a double-stranded piece of DNA. You'll notice that it's got the labels 3' and 5', 3' and 5'. So there they are. The dark blue strand is the top strand. The light blue strand is the bottom strand. So just pretend this is a double-stranded piece of DNA. And let's say that it's a double-stranded piece of DNA that I picked up at a crime scene. So it's just a random segment of DNA. But for a lot of the analysis things that we're going to learn next week to determine who the bad guy is, I don't need their entire DNA sequence. I just need a part of it. And I need lots and lots of copies of that part of it. And we'll talk about that in week 4, why you need lots of copies. And so let's just say that this part right here, this middle chunk, is the part that I need for whatever test I'm doing for the bad guy. So I don't need the entire segment, I just need this, and I need lots of copies of it. So we're going to use PCR on this piece to try and make lots of copies of just this middle piece and leave the rest behind. So there are 3 steps to PCR. So we're gonna put the starting number of DNA strands here in the middle, and we're going to, so the first step is to heat it up. Now, why would we heat it up? If you remember, in DNA replication, there was about 7 different enzymes that you guys have to memorize for DNA replication in the cell. There's one of them that unwinds the DNA strands, that's helicase. You've got primase that builds the primer. You've got topoisomerase that keeps the strands from getting too tight. You've got all sorts of things. Tons of these proteins, single-stranded binding proteins, all of them. And it turns out that enzymes can be purchased online. You can go online, you can buy enzymes, and scientists do it all the time, but they're really expensive. For a bottle like this big, that's got like an eighth of a teaspoon of enzyme in it, costs like $70 per enzyme. So it's really, really expensive, and doctors don't like to spend $500 just to get the enzymes, and then you have to get all the other equipment and the nice expensive PCR machines and stuff. So we try to keep the price down, believe it or not. We actually do try. And so the point of PCR is eliminate as many of those proteins as possible while still accomplishing the same thing. And thanks to this being in a test tube, we don't have to use the same rules that a cell has to. A cell, if you heat it up, it dies. We can't, so it has to work at body temperature in order to, and still divide the DNA. Here, we don't have to worry about killing the cell. We took the DNA out of the cell. And DNA is not alive, so we can't kill it. So it turns out when you heat something up really hot, it melts. DNA is the same thing. It can melt. If you remember from your week 2, DNA is structured as there are 2 backbone strands with the bases that are hydrogen bonded across the middle. Well, it turns out the backbones are covalently bonded. If you remember, that means they're really strong bonds. They're like glued together. And the bases are hydrogen bonded across the middle, which are really weak bonds, more like magnets sticking together. And so it's easier to break the middle bonds than it is to break the backbone bonds. And that's what we rely on here. When we heat it up to 95 degrees, so let's heat it up to 95 degrees here. This is in Celsius, by the way. So 95 degrees Celsius is pretty close to boiling for water. Water boils at 100 degrees Celsius. So just keep that in mind. We're at some pretty hot temperatures here. When you heat it up to 95 degrees Celsius, it turns out that that's enough to break the hydrogen bonds across the middle of the DNA helix, but not enough to break the backbone, which is good because that means the 2 strands separate, and we accomplished what helicase did with the help of topoisomerase without having to use either one. So I just separated the 2 strands without using 2 enzymes. I just saved myself $150 right there. Now let's move on. The next step in DNA replication was you needed primate. Once you got it unzipped, you had to have primase come in and build a little RNA primer. Because DNA polymerase 3 only knows how to make a strand longer. It doesn't know how to start one brand new. So you have to have primase in there. But remember, we're trying to eliminate enzymes. So what we do instead is we hop online, and there are companies that have really smart biochemists that work for them, where I can get online to their website and type in a little DNA sequence, type in a little sequence of for a primer and submit my order, and for like $5 they'll send me a tube of primers, pre-made primers. They'll build them for me and send them to me, and I dump them into my tube, and ta-da, I have primers. So ta-da, I just got rid of my primer. I don't need primase anymore because I just had a biochemist make it for me for $5 rather than $70 to buy the enzyme. Now, the catch here, people get this confused. In DNA replication, the primer was made of RNA. The reason for that is because for some random reason, God made primase so that it only knows how to work with RNA breaks. And primase is essential to build the primers in the cell. So it has to build with RNA, and then we have to come back in with DNA polymerase 1 and RNase H and cut the primers back out again so we can fill them in with DNA. Here in the cell, or here in the PCR, I'm having a biochemist in Ohio or someplace like that do it for me. And so he's not restricted to working with RNA bricks. So I just have him build it out of DNA bricks, and then I don't have to cut the primers back out again. So there, not only did I, by building it out of DNA bases, I'm not only eliminating primase, I'm eliminating DNA polymerase 1 and RNase H as well. So I just chopped out 3 more enzymes from my DNA replication process and saved a bunch of money. So the primers are made of DNA in PCR. They're made of RNA in cell replication. Okay, so the next step, because remember, we heated it up to 95 to separate the strands, and I need the primers that I've dumped in here. They're just floating around in this tube. I need them to be able to stick onto this other strand, but since it's so hot, they can't go back together again. So I have to cool it down to 55 degrees. So this first step, forgive me, I almost skipped this. The first step is called denaturing. Denaturing means when something is natured, that means it's together. That's it, right? It's its nature. It's in its natural form. So when you denature something, you undo its natural form. So the denaturing step is when we heat up the DNA to separate the 2 strands. That's step 1. Now we go to the second step, and that's called annealing. Annealing. The word anneal is a science term that means to stick together. So it's when, this is the step where we cool it down to 55 degrees and that allows the primers to go and stick to whatever DNA sequence on the template DNA strand matches the sequence of the primer. So we design our primers to stick to a certain spot in the DNA, if that makes sense. And traditionally, when we do this, we use 2 primers. So we make one primer that targets a certain sequence and another primer that targets a different sequence. And we have to pick these sequences very specifically. And I'll show you why here in a sec. But I'll show you the full reason why here in a sec. But the main rule to picking your primers, okay, let me go over the 3 rules for picking your primers here, because this is good for your homework. The 3 rules for picking your primers are, 1, let's come over here. So rule number 1, rules for picking primers. Thanks for putting up with me, guys. Rules for picking primers. Okay, rule number 1. The 2 primers you pick must bind to opposite strands. So for over here, remember, if I go back to when we separated them, there were 2 single strands of DNA. I don't want to throw in, if I throw in 2 primers that are on the same strand, that bind to the same strand, so they both stick to the top one and not the bottom one, it's not going to work. And I'll show you why here in a couple of minutes, hopefully. So that's what we mean. We have to have 1 primer that sticks to this top strand and 1 primer that sticks to this bottom strand. Good so far? Okay. I don't know why I ask those questions. You guys can't really respond, but it makes me feel better. Okay, here we go. The second rule is you want these 2 primers to be on opposite ends of your target sequence. So remember, so let me type out the rule. Opposite ends of target sequence. What we mean by that is, you remember in my little example, this middle chunk is the one that I'm trying to make photocopies of sorry guys, my brain is so dead today. Holy cow. It's the one that I'm trying to make tons of copies of to convict the bad guy. So I need to pick primers that, in addition to being on opposite strands, are on opposite sides of this thing. So I would want one on this side and one on this side. Does that make sense? Because this is that middle piece, I need one on either side of this segment in the middle in order to make it work. So that's the second rule. Because if you put them on both on the left side here, for example, it's not going to work. Okay. Rule number 3, the primers, let me think about this for a sec. The primers must be upstream of the target. Another way of putting that is when I build 5' to 3', because eventually we will build 5' to 3', and I'll get to that. If they're not upstream, if building 5' to 3', I don't copy the polymerase enzyme doesn't read the sequence of interest and copy it, we missed the mark. So if the polymerase, so if I pick a primer up here, for example, if I put a primer, if I put a primer right here, for example, then this would be the 5' end over here of the primer, and this would be the 3' end. So polymerase would hop on here and build that way. And that would not copy the middle piece that I want. And so that would make the PCR not work. So I need to make sure that the two primers, both of them, when you build 5' to 3', cross the area you want to amplify. You want to copy, I mean. Another way of putting that, and I'll just make it rule 4, is when you build 5' to 3', the primers must allow the polymerase to overlap. Meaning, I would want a primase over here that when it builds 5' to 3', it's going to go left to right. And if I put a primer here, building a 5' to 3', it's going to go over here. Now notice both of those overlap on this segment. Actually, they technically overlap on this. Does that make sense? Whereas if I put them on the other spots, if I put it over here, this one would build 5' to 3' going that way, and this one would build 5' to 3' going this way, and they wouldn't overlap, so I wouldn't get anything in the end. So building 5' to 3', the primers must overlap. Otherwise it won't work. That's basically the same thing as rule 3, but just phrased in a different way. So those are your rules for picking primers. And you can use those on your homework. Now, sorry, back to the process here. So we we've designed our primers well in this case. So we cooled it down to 55 degrees. Now notice this was our target sequence here in the middle. And I have one primer that's binding up here, so upstream of the target, and another primer that's binding on this strand that's going to bind upstream of the target. Now, we have to make these strands longer. Now, if you remember, in DNA replication, we used an enzyme called DNA polymerase 3. And DNA polymerase 3 bound onto the primer and read the template strand in the 3' to 5' direction and built the new strand in the 5' to 3' direction. Well, it turns out that scientists have yet to figure out a way to replace DNA polymerase 3. So we have to use DNA polymerase 3. That's the one enzyme in the whole setup that we keep. Otherwise, we throw all those other ones out the window. It's just DNA polymerase 3. And so this next step is called elongation, because the DNA polymerase 3 enzymes that we just buy them, you can buy them online, and we dump them into the tube. And when we heat it back up to 72 degrees, that's DNA polymerase's best, that's its home field. Then they stick to the primers and they build in the 5' to 3' direction. Exactly like we did with DNA replication. They bind onto the primer and they build in the 5' to 3' direction. Now notice these ones overlapped right there in the middle. This one built 5' to 3' going that way, and this one built 5' to 3' going that way, and they overlapped, and both of them copied the segment I'm interested in. That's good. That means we designed our primers right. Whereas, again, like we talked about, if they were in the other spots, they wouldn't overlap and it would be bad. Okay, so that's that. Now, the interesting thing about this, so again, this one's called elongation because we're sticking to the primer and we're making it longer. The interesting thing about this is we don't just use any everyday average DNA polymerase 3. We use a special one, and let me pull up a picture of it quick here. Or, sorry, let me pull up some pictures here. Give me a sec. Okay, here we go. So this particular DNA polymerase 3 is called TAC polymerase, T-A-Q polymerase, and we pronounce it TAC, like tacky. It's called TAC polymerase, and the reason it's called TAC polymerase is we took it from this bacteria. Bacteria use DNA polymerase 3 just like humans do, and this bacteria is called Thermus aquaticus. Thermus aquaticus, and so the T comes from thermus, and the AQ comes from aquaticus. So this just says it's a DNA polymerase that we took from Thermus aquaticus. So that's what the TAC means, TAC polymerase. So it's just DNA polymerase 3. It's exactly the same thing. The one difference is it's got this slight modification to it that makes it very helpful for PCR. Remember, in PCR, we have to heat this thing up to 95 degrees. Most things fall apart at 95 degrees, especially in the body. So if we threw in a human DNA polymerase into our PCR test tube to do elongation, it would actually fall apart in the denaturation step and not work in the elongation step because it would be all melted. It would be broken. Proteins are much more complicated, much more structured than DNA is. So DNA can just come right back together again, but proteins, once they're denatured, it's really difficult to get them to go back together again. So we try to avoid that. And it turns out that this bacteria has the solution. It lives in Yellowstone. They actually found it. This is the exact lake where they first discovered it. It's a hot spring in Yellowstone. It's called Mushroom Spring, and it's really, really hot. If you've ever been to Yellowstone, you'll notice, I mean, the water, like steam is coming off right there. Like this water is close to boiling. And they've got those beautiful lakes that have the orange and stuff like that around the outside edges. That's bacteria. So there are bacteria, most bacteria die when you heat them up. That's why you boil your food. But not all of them do. In fact, some of them have adapted to be quite happy there. And Thermus aquaticus is quite happy living in this lake that's near boiling. But in order to live there, it needs to make sure its proteins won't fall apart at near boiling temperatures. And so it's got modifications to the structure of its enzymes that make them not fall apart at near boiling temperatures, and they still do their job. So by taking the polymerase from this Thermus aquaticus, we can dump it into the tube, go through denaturation, and the DNA will fall apart, but the polymerase won't. And then we cool it down and do the annealing and warm it back up for elongation, and the polymerase is ready to go because it didn't fall apart. That's why we use TAC polymerase, because it's heat resistant. It won't melt at the high temperatures we need to separate the two strands. Kind of fun, huh? So yeah, there's that. Now let's go back to my simulator here. Okay, so we started out with one double-stranded piece of DNA, and now we've got two. Woohoo! You know, we doubled the amount of DNA strands, but we're trying to make thousands of copies. Two, on a DNA level, 2 copies isn't much better than 1. But we just finished the DNA replication process. However, watch this. You remember, with DNA replication in the cell, you have you have Okazaki fragments on the reverse strand because you bind onto a primer and you build that way, but everything behind the primer didn't get built. Notice here's my primer. Everything before it didn't get copied. And we're okay with that. We leave it. Because remember, I'm not trying to copy the entire chromosome like a cell is. I'm just trying to copy one little piece. So I don't need to copy everything. I just copy what's in between the 2 primers and forget everything that's before them. That's going to be important here in a sec when we talk about amplified products. The other thing that's important to know about elongation is, in addition to adding the DNA polymerase, we add in these little pieces called dntps. And let me even pull up a little Word document and address those. So we add in these little pieces called dntps. Now the dntps, it's an acronym, as you might suspect. We love our acronyms in science, so get used to it. The D stands for deoxyribose. This stands for nucleoside, forgive me, nucleoside triphosphate. What these are so it's deoxyribose nucleoside triphosphate, or nucleotide triphosphate. You can get away with it either way. What these are is individual DNA bases. It's the phosphate, the sugar, and the base. Holy cow, my brain just short-circuited. But let me show you a picture here. Okay, so here's your standard. Here's just like your general layout of a DNA base that you learned about last week. You've got your nitrogenous base. This is the A, C, G, or T. You got your pentose sugar right here, and then the phosphate, and they're linked together like that. Well, this is your standard base. The difference between this and a dntp is there's 3 phosphates right in a row. So we've got the base, the sugar, and 3 phosphates, one hooked to the next. So it looks kind of like this over here. So this is your standard adenine base. This is just the chemical structure of it. You got your phosphate over here, your pentagon here in the middle, and your adenine nitrogenous base over here. This is what we call adenosine triphosphate, ATP. There's your adenine right there. There's your pentose sugar. It's the same thing, but instead of having 1 phosphate, we just got 3 of them hooked right together right in a row. This is a dntp. So the dntps are the A's, C's, G's, and T's, just the individual building blocks. We use these 2 extra phosphates as the enzymes use the 2 extra phosphates to link the 3rd phosphate to the next base. But if you want to learn about that, you don't have to know about it. But if you want to learn about that, you can go to my Tutor Files webpage and find my article, Why Can DNA Polymerase 3 Only Add Nucleotides in the 5' to 3' Direction, if you're curious. It's a lot of fun. So anyway, so that's what the dntps are, is they're deoxyribose nucleosides, or nucleotides. I'll just switch that back to a T, nucleotides. For this class, nucleosides and nucleotides are basically the same thing. Deoxyribose nucleotide, so it's back over here. So it's a deoxyribose nucleotide triphosphate, 3 phosphates. So it's a deoxyribose nitrogenous base with 3 phosphates, hence the name dntps. That was more than you wanted to know. I'm sorry, I'm rambling today. Okay, let's get back here. Okay, here we go. Finally, we'll move on. I promise, guys, I'm almost over. I'm very long-winded. Okay, so we've got 2 strands. So now we're going to do the process again. So we start back at the beginning, but this time, so those 3 steps, the denaturation, annealing, elongation, those make up 1 round of PCR. And each time you go through a round, you double the amount of DNA that you started with, because you separate the 2 strands and you copy them both, so you double it. So we went from 1 to 2. Now we're going to start the 2nd round, which means we go to denaturation again. So we're going to heat it back up to 95 degrees, and both of those strands are going to separate. Now, I apologize if this is confusing. This strand here used to be this one right here. And this strand right here used to be this one right here. We've just separated them, and for the sakes of my little program here, they're now 4 separate individual strands, and I moved these ones down here. I apologize for that, just don't be lost. So the strands that we built are now new template strands. Then we go through annealing, stick the primers on, and they're always going to stick on the upstream side. It's the same primers because it's the same DNA sequence. So they stick on the upstream side, and we've got, so this one sticks here and there and here and there. And then we do the elongation phase, where DNA polymerase 3 comes in and uses the dntps and elongates them in the 5' to 3' direction. So now, since I've gone through 2 rounds of PCR, we have 4 strands, 4 double strands of DNA. So because we doubled it both times. Then we do it again. We heat it up to 95 degrees. Now we've got 8 separate individual strands because all 4 of them separate to become template strands. So we have 8 new template strands. And then we anneal again. We drop it back down to 55 and they stick on there. And then we elongate. Now notice here, this is where things start to get interesting. And I promise this leads into the second half of my video, into the amplified products. Notice here, if I back up one step here at the annealing, on round 3, here, this bound in the 5' end, right? Same with this one. It binds on the 5' end of this new strand. So the 3' end of the template strand, the 5' end, it sticks right here. And it's going to build 5' to 3' just as normal. But look at this. It ran out. Now, why is this strand shorter? We didn't cut it. But remember, up here, this one used to be the strand that came from here. And so we just simply didn't cut it or copy it before the primer. And so essentially, we clipped it off. So the DNA polymerase, it only knows how to read a template strand and build new bases. So when I elongate this one, this particular one, it's going to hop on here, build 5' to 3', and, oh, sorry, this one right here, it's going to build 5' to 3' and run out of tracks. And it's just going to stop right there. Now notice, this strand is now only this long because we're not copying anything before here, and we ran out of tracks over here. So it's just that long. Same with this one down here. This is where things get interesting. I'm coming to a point with this, guys. Okay, here we go. Denature. So now this is round 4. This is where things get interesting. We denature, we add on the primers, and we elongate. Now, look here at these bottom ones. When we separated back here on denaturation, there were 4 of these strands that had been clipped off on both ends because we didn't copy anything before the primers, and after several rounds, that makes them only so long. And then when we elongate them, we come out with 4 double-stranded segments that are only this long. And every time I go after it, all of these strands up here will continue to produce more of these, 4 of them every round. And these 4 will double every single time. So in essence, these weird ones up here that are all different lengths, we'll keep the same number of them no matter how many rounds of PCR we do, and we're going to make a 10 bajillion of these shorter segments. Hence, this is the one that is amplified. That's your amplified product. A product is is the result of a process. And so, yes, we've got like, what, 14 of these weird strands, but in 14 weird strands in 1,000, it's the other than 14 ones that were amplified. So this is your amplified product. This section of DNA right here is your amplified product. Now, how do you determine that? Well, remember what determined how it got to this long. It was from the edge of one primer to the edge of the other. That's your amplified product. The length of your amplified product is the length of the left primer plus the length of the piece in between the 2 primers plus the length of the right primer. And that's going to be the length of your amplified product, because that's where the term comes from, is this is that piece. So it's primer plus middle piece equals amplified product. If you just do that math, your homework's going to be a piece of cake. That's what the amplified product is, and that's how you find the length of it. Primer plus everything in between. And if you did it right, this piece in between is the segment that you're looking for, and then we can use it for any of those processes at the beginning. So there you go. That's PCR and how to find out the amplified product. Sorry, this was a long video. Hopefully it was helpful. Please email me at dallin.stokes17@gmail.com if you have any questions, or if there's anything else I can help. Please feel free to set up a tutoring appointment, and we can go over this again. And there's also my tutor files webpage, my DNA tutor files webpage that I send out right here. That's got a lot of other resources as well. So hopefully this helps, and have a wonderful day.